In the lessons from our previous chapter, we talked in depth about known protein structures and how they relate to their functions. In this chapter, we want to look at the methods that allow us to extract and isolate a target protein so that we can perform those types of studies. Our first job is to isolate our target protein from the hundreds of proteins present in a single cell. This will mean eliminating many contaminants in order to obtain a pure sample. Of course, we must first rupture the cell in order to access the proteins within. Homogenization is a common technique for breaking cells open. Pictured here is the process of cell rupture using a Potter-Elgin homogenizer. It contains a thick-walled tube into which the solution of cells is placed. A tight-fitting plunger is then pushed through the tube, thereby breaking the cell membranes. This mixture is then strained to remove extraneous tissue and cellular structures. Note that the proteins are fully soluble in the liquid fraction. Ruptured cells can then be subjected to differential centrifugation. The sample is spun at high speeds to precipitate solids, and the force of gravity is increased with each spin. With each increase in force, various cellular components are extracted, as illustrated here. A more straightforward approach is simply to grind the tissue in a blender in a suitable buffer. Another option is to use a device known as a sonicator, which uses sound waves to rupture the cell membrane. Once the cellular extract has been separated from the membrane fraction and collected, protein purification can begin. We'll consider the early stages of purification in our next lesson.